Okay, so like I just said, all cells will undergo the process of glycolysis, whether oxygen is present or not. And that's once again where we are taking glucose, and in the process of glycolysis in the cytoplasm, we're going to break glucose down into two 3-carbon pyruvate molecules. Okay, and in that process, we will have created 2 ATP via substrate level phosphorylation. Well, actually 4 ATP, but 2 net ATP, because remember, we had to invest 2 ATP, and also 2 NADH molecules, which are carrying those high energy electrons. So now we're sitting out there in the cytosol. The pyruvates are kind of floating around. Hey, what do we do? Well, that all depends upon whether or not oxygen is present. If oxygen is present, then the pyruvates will be transported into the mitochondria and undergo the process of the Krebs cycle to further extract the energy out of those pyruvate molecules. If, however, there is no oxygen present or not enough oxygen present, then the pyruvates will undergo fermentation in order to allow for glycolysis to continue. And we're going to talk about two major different types of fermentation. One is ethanol, which will result in the production of alcohol, and the next one is lactate, which is going to result in the production of lactic acid, which is done mostly by animals, and that's what will result in muscle cramps. So the key event is oxygen present? Is a two-step process. We go to pyruvate, then to acetaldehyde, then to ethanol, whereas the lactic acid fermentation is a one-step process. We go to the pyruvate, and then we go directly into lactate or lactic acid. Also notice that there is no CO2 produced in this process. Okay, so there's a couple of the main differences that occur. And both lactic acid and alcohol are kind of like poisons for the cell. Okay, the cell can't really tolerate high concentrations of these, and if the concentrations get too high, then the cells will stop producing glycolysis and they will no longer have energy and they stop undergoing this process. And that's the reason why sprinters have to stop and rest and breathe and get rid of the lactic acid. Okay, humans can't really sprint for more than about 800 meters in an all-out sprint because their bodies are just can't handle that. I also want to talk a little bit about negative feedback regulation. Okay, why would the body want to make more ATP or an organism make more ATP if there is already enough to go around? It would be a waste of energy and resources. Okay, so negative feedback inhibition or negative feedback is basically when the products of a reaction are going to somehow inhibit the production of more of the same products. And if you look at this diagram up here, what we see is the process of cellular respiration with the three major steps. We see glycolysis, then we see the Krebs cycle, and we see the electron transport chain and oxidative phosphorylation. Those are the three major steps of cell respiration: glycolysis, Krebs cycle, electron transport chain. Okay, and as you can see, ATP is going to come in. Once enough ATP is made, it's actually going to stop the production of more ATP in glycolysis. And that phosphofructokinase, that's an enzyme, ASE, that's the key stepping point. And that's actually the glycolysis phase of glycolysis. That's the sugar splitting phase. And here's a better picture of the phosphofructokinase. As you can see, once the phosphofructokinase phosphorylates the fructose 6-phosphate, that'll make fructose 1,6-bisphosphate, which will then split into 2 3-carbon molecules, which will eventually become pyruvate molecules. Okay, so that's the energy investment phase. And enough ATP will dock into phosphofructokinase and basically change the active site of that particular enzyme. So ATP is acting as an allosteric inhibitor to stop the production of more ATP. And again, that is negative feedback inhibition. Positive feedback would be that the production of ATP would lead to more ATP and more ATP and more ATP and so on and so on and so on. So that would not be very beneficial to an organism. So the last thing I want to talk about is how do we measure the processes of photosynthesis, cell respiration, or fermentation? And as I've done in demonstrations in class, look at the equations. For photosynthesis, we could measure the amount of CO2 that is being absorbed, the disappearance of CO2, or we could measure the amount of oxygen being released. We could also potentially measure the amount of sugar, although that would be a little bit harder. The easiest thing for us to do in our lab would be to measure the disappearance of CO2 using a CO2 probe. And for cell respiration, the same thing. We could look at several different things with cell respiration. We could look at the utilization of oxygen on the reactant side, and on the product side we could look at the production of CO2. That would be the easiest thing to measure. We could potentially also measure the amount of heat being released. That would be another another idea, or excuse me, another thing we could measure. And with fermentation, the easiest thing to measure would be the amount of lactic acid or ethanol being produced, and or potentially looking at the amount of sugar being consumed. So we could look at the amount of glucose being consumed or the amount of lactic acid or ethanol being produced. Okay, so those would be some of the easiest ways to measure how or I should say, the rates of either photosynthesis, cell respiration, or fermentation. And that is it for today. Hopefully you'll do well in the test.